Pharma.
Welcome back to our channel. In today's video, we'll be diving into the fascinating world of molecular diagnostics and protein therapeutics. So grab a cup of coffee and let's get started. Molecular diagnostics play a crucial role in modern medicine. These diagnostic techniques can detect specific molecules with high sensitivity and accuracy. There are two main categories of molecular diagnostics: immunological diagnostic procedures and nucleic acid diagnostic systems. Immunological diagnostic procedures rely on the specificity of antibodies for particular antigens. One commonly used technique is the enzyme-linked immunosorbent assay, or ELISA. This test can detect proteins, viruses, bacteria, and even low molecular weight compounds. It can be performed using either a direct or indirect approach. In the indirect approach, a primary antibody is added to the test sample, followed by a secondary antibody that binds specifically to the primary antibody. This secondary antibody is linked to an enzyme that can convert a colorless substrate into a colored product, allowing us to observe or measure the amount of the target molecule. Monoclonal antibodies are essential tools in immunological diagnostics. They are produced by fusing B lymphocytes with myeloma cells, creating hybrid cells capable of both replication and antibody production. These hybrid cells, known as hybridomas, are selected using a technique called the HAT procedure. Nucleic acid diagnostic systems utilize nucleic acid hybridization, or PCR, to detect specific DNA or RNA sequences. PCR, or polymerase chain reaction, is a technique that amplifies target sequences, increasing the sensitivity of the test. This allows for the detection of even minute levels of the target molecule. To develop a DNA hybridization probe, the DNA from a pathogenic organism is cut with a restriction enzyme and cloned into a plasmid vector. These recombinant plasmids are then screened with genomic DNA from both pathogenic and non-pathogenic strains. The plasmids that contain sequences specific to the pathogenic strain become the basis for species/strain-specific probes. Non-radioactive hybridization procedures have revolutionized diagnostic testing. These systems use enzymatic conversion of chromogenic or chemiluminescent substrates for signal amplification. Biotin-labeled nucleotides are incorporated into the DNA probe, which binds to the target DNA. Streptavidin then binds to the biotin molecules, and biotin-labeled alkaline phosphatase binds to streptavidin. Finally, alkaline phosphatase converts the substrate into a chromogenic or chemiluminescent product. Now, let's shift our focus to protein therapeutics. These are therapeutic agents derived from proteins that can be used to treat various diseases. One example is human growth hormone, or HGH. HGH was one of the first therapeutic proteins to be approved for human use. However, it had a short half-life and caused side effects due to binding to prolactin receptors. Scientists developed solutions to these challenges by modifying the HGH protein. One solution was to fuse the extracellular domain of the HGH receptor to HGH, resulting in a protein with a strong tendency to dimerize. This extended the protein's half-life and improved its efficacy. Another solution involved fusing the C-terminal end of HGH with the N-terminal end of human serum albumin, creating a fusion protein called albutropin. This fusion protein has a longer half-life and is currently undergoing clinical trials. Hybrid human-mouse monoclonal antibodies are another exciting development in protein therapeutics. These antibodies can be coupled with drugs, prodrugs, or enzymes and specifically target proteins found on the surfaces of certain cells, such as tumor cells. To make these antibodies more compatible with the human immune system, the complementarity determining regions, CDRs, of rodent monoclonal antibodies are substituted into human antibodies. And there you have it. A glimpse into the world of molecular diagnostics and protein therapeutics. These advancements have revolutionized the field of medicine, allowing for more accurate diagnoses and targeted treatments. If you enjoyed this video, don't forget to hit that like button and subscribe to our channel for more exciting content. Thanks for watching.
Pharma.